Hello students, last time we had completed Phycomycetes, under which we had completed Oomycetes and Zygomycetes, and now we are on the next class, that is class Ascomycetes. Ascomycetes are also called as the sac fungi. Why they are called as sac fungi? You will tell me yourself when we learn about their life cycle. Which fungi are included in the class Ascomycetes? The baker's yeast, Saccharomyces cerevisiae, belongs to this class. The fungus Penicillium, from which you obtain the antibiotic penicillin, belongs to this class. Another one, Aspergillus, which is known to cause infestation of stored food grains, belongs to this class. There are other examples as well. Have you heard of morels and truffles? They are similar to mushrooms, but the ones that we eat commonly, they belong to Basidiomycetes. Morels and truffles are also similar type of mushrooms. They belong to class Ascomycetes. They are delicacies in Western countries. Okay, let us write down about them. Habitat. They are mainly saprophytic, so they are decomposers. There are also parasitic species, and many forms are known to be coprophilous. Coprophilous means dung-loving. Cell wall. Cell wall will be made up of chitin. Chitin cell wall had started from Zygomycetes itself. Next, mycelium. Septate or aseptate, septate mycelium, that will be branched. Asexual spores. Asexual spores, if you remember, while we were doing the asexual spores, we had come across one conidia. Under conidia, the examples that you have cited, which is Penicillium and Aspergillus, they belong to this class itself. This class is known to produce only conidia as their asexual spores. Conidia. Sexual spores. Sexual spores will be ascospores. We will see how ascospores are produced in their life cycle. These will be haploid spores. Examples. Tell me one by one the different yeasts. Example, baker's yeast, species name of which is Saccharomyces cerevisiae, belongs to this class. Then penicillium, which produces the antibiotic penicillin. Next one, Aspergillus. Then there is one called Claviceps. There is one called as Morchella. Morchella produces morels, which is a fruiting body. It's a delicacy. Then there is one called Tuber, which produces truffles, which are also known to be a delicacy. Next, we are going to look at the life cycle of Ascomycetes, in which we will see how the ascospores and all will be produced. We are looking at the life cycle of Aspergillus, which is a black colored fungus. By the way, have you seen penicillium? The patches of penicillium are seen on citrus fruits which have been kept in the open for too long. They will have patches, green colored patches on them. They will be of penicillium. Similarly, Aspergillus is a black colored mold. So let's start with the life cycle. So as you can see, there are two fungal hyphae here, denoted as plus and minus. What do they represent? Compatible mating types are close to each other. These that you can see here, these are their sex organs. For the male, the sex organ is called antheridium, and the female sex organ is called ascogonium. And now they are about to undergo the first step of sexual reproduction, that is plasmogamy. Let's find out what happens after plasmogamy. Can you tell me your observations post plasmogamy here? What is happening? As you can see, the antheridium has fused with the ascogonium. The nuclei from the antheridium have entered into the ascogonium, as you can see, and from the ascogonium are arising new type of fungal hyphae over here. And if you look carefully, you will see that these hyphae are now bearing both the types of nuclei of the parents. What is happening here? Is it what it is supposed to happen? They were monokaryotic before the plasmogamy, and after plasmogamy has happened, new type of fungal hyphae that are growing are having two nuclei in each. These are the dikaryotic hyphae. Isn't this supposed to be in the higher fungi? Since these are higher fungi, you remember? What happens in the higher fungi? Plasmogamy is not immediately followed by karyogamy. They go through a phase called as the dikaryo phase. So this is what is happening. So the hyphae that are going to grow, they will be dikaryotic hyphae N and N. So these will continue to grow. Next, what is going to happen is these dikaryotic hyphae will keep growing and they will keep on clumping together. Clumping of dikaryotic hyphae. Once the clumping happens, it results in the formation of different types of structures which are called as the fruiting bodies. In the previous lectures, we saw how the clumping of the dikaryotic hyphae leads to the formation of the mushroom. The mushroom is nothing but a composition of dikaryotic hyphae, of basidiomycetes. Right now we are in Ascomycetes. In these fungi as well, the clumping of the dikaryotic hyphae result in a mushroom-like structure of different shapes in the different species of Ascomycetes. Different types of fruiting bodies are formed. For Aspergillus, the fruiting body does not look like a mushroom. It looks like a small mustard seed sort of. The structure will be made up of dikaryotic hyphae. Inside also there will be dikaryotic hyphae. So this is the side view. This is a fruiting body. The fruiting body of Ascomycetes is called as ascocarp, wherein carp means fruit. Ascocarp means fruiting body belonging to Ascomycetes. What will be the fruiting body of mushrooms called that we eat? That will be called as, mushroom will be called as a basidiocarp. Okay, we will see when we are doing basidiomycetes. You can still see the dikaryotic hyphae inside this fruiting body. Let us analyze one of these hypha. So one dikaryotic hypha I am keeping here. They are still in their dikaryo phase. Karyogamy has not yet happened in them yet. It is going to happen now. Look, what is happening? What has happened here? This terminal tip of this dikaryotic hyphae has curved downwards like this, in this manner, to form a hook sort of. This is called as a crozier formation. Crozier formation. This is Ames related terminology. This is called as the terminal cell here. What is with the terminal cell? Let's find out. See what has happened in the terminal cell. Both the N and N nuclei of this terminal cell have fused together to form one nucleus that will be a 2N nucleus. So finally in this terminal cell we can say that karyogamy has happened. What is the next step after karyogamy in sexual reproduction? Meiosis, which will happen right there. Meiosis is going to happen in the terminal cell. 4 nuclei will be produced, all of N and N ploidy. Meiosis has happened. Does it stop at meiosis? Look what is happening. What has happened to this terminal cell now? Look, it has got turned into a sac-like structure here, which is called as the ascus. And inside the ascus, 8 different nuclei will be seen. The next step that precedes meiosis will be one step of mitosis. There were 4 nuclei in this structure post-meiosis. Now it will result in the formation of 8 nuclei, because each of these nuclei has undergone mitosis. The terminal cell has got converted into a sac-like structure over here to contain these 8 nuclei. That sac-like structure is called as the ascus. Inside this fruiting body you will find several such asci. And what has formed of those 8 nuclei is they have got converted into ascospores. These are the ones, ascospores, the sexual spores which will be contained in the fruiting bodies. Ascus is singular, plural is called asci. As such kind of sac-like structures will be present in the fruiting bodies, and then the mustard seed-like looking fruiting bodies are going to eventually desiccate and disperse the ascospores outside, which will be carried by the wind. They will be sexual spores. They will not be conidia, the asexual spores. They will be sexual spores, resulting in formation of fungal colonies of different genetic variability from the parents. Can you tell me why these fungi are called as the sac fungi? Because they produce such sac-like structures which are called as the asci. If you notice the ascospores, how will you describe the ascospores? These will be endogenously or exogenously produced. They are endogenously produced. And how many in number are they? They are 8 in number per ascus. Ascospores will be endogenous origin and always 8 in number. So this is the life cycle of Aspergillus, which is a common Ascomycete. And in the life cycle, we have learnt the 3 steps of their sexual reproduction: plasmogamy, karyogamy, which will be delayed. As you can see, it's delayed, it's not immediate after plasmogamy, as in the lower fungi. A dikaryophase, a prolonged dikaryophase is seen in them. They are higher fungi, that's why. And then finally there is karyogamy and meiosis as well. Another step of mitosis also takes place before the sexual spores are formed. These are their sexual spores, ascospores. This is ascus, not to be confused with ascocarp. The fruiting body is called ascocarp. This is not a fruiting body. This is a spore-bearing structure. I would like to mention one more example of Ascomycetes, that will be Neurospora crassa. Neurospora crassa is used for biochemical and genetic engineering research work. It's a pink colored Ascomycete. Penicillium is green colored, Aspergillus is black colored, Neurospora crassa is pink colored mold. These colors that they get, is because of the conidia that they produce. This one produces pink colored conidia, Neurospora crassa, pink mold. Important example, used for genetic engineering and biochemical research. So that is all about the Ascomycetes.
महादेव के नाम पे सब्सक्राइब कर दे।